and also Shoba, who
Hi,
I worked with.
Just for context, Eddie was one of our fellows. I won't say when, but we've followed his career since, and it's great to see you today.
Wonderful. Panelists, by the way, throughout the panel, if you want to respond or add on to anything that one of your fellow panelists says in response to any of the questions, please feel free to wave at me in your screen or chat to me, and I'll be happy to toss it to you after the other panelists is done with their response if a question feels like something that you'd like to weigh in on as well. I'm going to be adding our YouTube live stream to our website and starting the recording that we'll have going here in just a moment. One more reminder for all of the media on the call. We're going to have a recording and a transcript available of this call, so please do just keep your eyes out for that sometime early tomorrow morning for the transcript and video. And you're welcome to use any of the quotes and even clips of today's presentation in any of the work that you're doing to write about these topics. And we also would love to get your questions so that we can make sure the panelists give good quotes that you're going to find useful. So please do chat your questions to me. I will offer for you to ask the question yourself, or if you don't want to do that, I'll ask it for you. And we'll go ahead and get started in just a second. Welcome to this Newswise Live expert panel. Today we're talking about the COVID pandemic, the vaccine trials underway, and other related topics. I want to go ahead and get started because we do have several panelists that we want to get to, and I'd like to start by introducing Dr. Edward Jones-Lopez. I'll highlight your video, Dr. Jones-Lopez. He's assistant professor and an infectious disease specialist at Keck Medicine at the University of Southern California. So Dr. Jones-Lopez, you're working on the AstraZeneca trial there at USC. Last week, we heard that the FDA has authorized resumption of that trial after it was determined that a participant who died of causes unrelated to the vaccine. Can you tell us about that process for resuming the trial and what people should know about how this has been, a step to ensuring safety of the vaccine and what you think people should know about that process?
Sure. Thank you very much for inviting me to participate in this distinguished panel. As many people know, the AstraZeneca vaccine was paused about 6 weeks ago, and because of a side effect that occurred in the UK. And that pause was an international pause, and it was fortunately lifted last week, I believe it is, on Friday. Before that, it had been lifted already in the UK, in Brazil, and in South Africa, which are the other 4 countries where this study is being conducted. So as of the next week, probably, there's going to be some changes to the consent form, obviously, and when those are approved, as of next week, probably, if not the week after that, enrollment into the trial will be resumed after the pause that occurred again about 6 weeks ago.
Thank you, Dr. Jones-Lopez. I want to go next to Dr. Robert Salata. Dr. Salata is professor of medicine and also epidemiology and international health at university hospitals. And from Cleveland Medical Center, Dr. Salata, you are involved in the Pfizer trial. Can you update us on the progress with enrollment in that trial, how many volunteers there are, and what you're doing at University Hospitals Cleveland Medical Center?
Thank you. Also, I want to appreciate the fact that several of my colleagues are on the panel as well. To add one thing to what Eddie Jones-Lopez just said, that patient in the UK did not die. That occurred in the Johnson & Johnson vaccine trial, but that side effect was called transverse myelitis. But they determined, including very carefully here at our FDA and the NIH, which is co-sponsoring that study, that this was not shown to be definitively related to the vaccine. The Pfizer study is different. It is a collaborative process between Pfizer, one of the largest pharmaceutical companies in the world, along with a German company called BioNTech. And it is unlike the AstraZeneca vaccine, which has another virus called adenovirus, a chimpanzee one, which is coupled to the coronavirus genes. In this case, it is something called messenger RNA, which we all have, and it's coupled to the spike protein. That is the projection that you see in all pictures of coronavirus. And that is the target, we think, for developing an effective immune response in the end, as had been shown in earlier studies of SARS-1 in the early 2000s and time period, and also MERS, the Middle Eastern respiratory virus as well. And in preliminary studies, including the AstraZeneca vaccine and this one, in monkey models, it was antibodies that developed against that spike protein that were protective once vaccinated and then challenged with the infectious agent. So we have been working on the Pfizer vaccine. We will be participating as well in AstraZeneca, which for us is just opening. But we started to vaccinate folks in the late part of August. And as of this week, we have halted further enrollment at our site. We have enrolled 180 subjects to date. There are 120 places around the world that are participating. We're the only one in Northeastern Ohio that we're aware of. And we have not seen any major safety signals in the Pfizer vaccine trials to date. And in the preliminary studies that were already published, there really is a very robust immune response to this particular vaccine. And it has been shown both in terms of antibody response, which everybody has heard about, and it was 5 to 10 times higher that we saw in natural infection. And we're looking to see about the longevity or the sustainability of that antibody response. But also, we also looked at T-cell responses, another form of immune response, which we think will be important as well. That said, Pfizer is closing in on their intended recruitment target of 44,000. The only group that will be vaccinated into November will be a group of children between the ages of 12 and 17. And why was that done? Well, many advocates had pushed forward with that. And when we get to the point of general distribution of vaccine in the United States and other countries, we want to know that it's also safe and effective in children. There will be.
Thank you, Dr. Salata.
Yeah, no
we
problem.
haven't heard yet about the younger adults yet being in any of these trials. That's very interesting. Thank you.
Yeah, no problem.
I want to go next to introduce Dr. Stephen Spector from UCSD. Dr. Spector is a professor of pediatrics at UC San Diego, and they are working on the Moderna trial there at UCSD. Dr. Spector, can you tell us a little bit about that? The Moderna trial has been reported to be coming along very quickly. What stage of things are you at in terms of administering the doses to all the volunteers, and what can you tell us?
Right. Well, similar to the Pfizer vaccine, the Moderna vaccine is also a messenger RNA vaccine that's in a lipid nanoparticle coat, so that that nanoparticle coat, one, helps the RNA to get into the muscle, and two, serves as an adjuvant to induce the immune response. I think that this particular vaccine has gone through very similar studies as what you've heard in the preclinical aspect for the Pfizer study, and as well as AstraZeneca and the other vaccines that you'll hear about. Moderna's trial has actually completed enrollment of the 30,000 participants. And in fact, last Friday, it closed enrollment at approximately 30,400 study participants. I think importantly, there was a major effort to expand the demographics of those individuals who participated in the study. In the end, overall in the study, there was about 67% whites, 20% Hispanic, Latino, about 4.5% Asian, and a little over 9% African American and Black. I think the other thing that was very important about the study was that many sites, such as our own, tried to bring a demographic into the study that represents the community that we live in. And so, for example, here in San Diego, where there's a lot larger Hispanic population of about 34%, we were able to enroll 38% of Hispanic Latino into the study, about 3.5% African American or Black. We have about a 4.5% percentage in San Diego. And then I think importantly, we actually enroll less than 50% of our population in whites. So it's representative of our community, and hopefully that if this vaccine or any other vaccine is licensed, we can say that this vaccine has efficacy and safety in a broad, for broadly in people, regardless of their ethnic or demographic backgrounds.
Thank you, Dr. Spector. I want to introduce next Dr. Shobha Swaminathan. She's an associate professor at Rutgers, New Jersey Medical School, and she's the principal investigator for the Moderna trial at that site. So similar to Dr. Spector, you're working on the same vaccine trial. We talked about how that's gotten to the full enrollment. Tell us a little bit about how that's going at Rutgers, and especially the importance of those underrepresented racial and ethnic minorities in your region.
Thanks so much, Tom. So as Dr. Spector just mentioned, the Moderna vaccine trial has completed enrollment. At Rutgers, New Jersey Medical School, we didn't start enrollment as soon as many of the other sites. That being said, we enrolled 57 participants, and at New Jersey Medical School, the reason we took our time is what, as Dr. Spector mentioned, representation of populations most vulnerable to this pandemic is critical to ensure that the vaccine, once approved, is actually proven to be safe and effective in those populations. But that also requires a lot of community engagement activities to get the community's trust and buy-in. So we've had to engage our community in multiple different sessions to increase awareness about these trials, to emphasize the safety measures that are in place, because at the end, once these vaccines are approved, we want to make sure that there is public trust in these vaccines, because nothing will be effective if the people don't take it. So at our site, of the participants enrolled, 70% were of different minorities, including Black and Hispanic, and about 25%, in fact, less than 25% were white, because in the second half of the enrollment, Moderna made a big push to ensure that the clinical trial population was at least as representative as possible of the general US population, so that the results would be applicable to the entire country.
Thank you, Dr. Swaminathan. Next, I want to introduce Dr. Perry Halkitis. He's Dean of the Rutgers School of Public Health. Dr. Halkitis, we've heard from all of these great doctors participating in these vaccine trials. As a public health expert, let's talk a little bit about where we are with the pandemic. We're seeing record numbers of cases, some areas around the country reaching ICU capacity. There's a lot of concern about this going in the wrong direction. What do you feel is driving these numbers, and what needs to be done?
Right. So thank you for that question, Tom, and thank you for the opportunity to be here and to share some ideas with my colleagues. I appreciate all of the efforts that are going on in developing the vaccines throughout the country and making sure that we have representation across the population so the vaccines work. I want to add one caveat to everything that's been said so far, which is these vaccines will only work if people trust them and if people actually take them. Right. So we have as much work to do on the behavioral aspect of vaccine as we do on the biological aspects of vaccine. So having said that, I think what we've seen over the course of the last several months, in fact, throughout the course of this pandemic, is a lack of behavioral maintenance. Public health folks know that human beings have a very difficult time maintaining their behaviors, whether it's their 7 days of their antibiotics or 5 days of their gym or eating on a regular basis good food. People have a hard time maintaining behaviors. And so the term COVID fatigue has been used. I don't really like that term myself. I just think it's just people not following their behavioral patterns. That I think is ultimately driven from the work that we've done here at Rutgers School of Public Health, in part by a lack of personal responsibility and altruism. I think it is very easy to think of oneself as being immune to this disease without consideration to other human beings who might be more susceptible to the disease. The actual things that have perpetuated the virus and the spreading of the virus has been, for the most part, informal social gatherings or formal social gatherings for that matter. I think that the restaurants and the gyms have been really careful about maintaining environments that are somewhat safe. But individual gatherings in people's homes, in people's backyards, in people's pools have been the factors that have perpetuated this disease. We also see the record highs in part because more and more younger people 18 to 39 are becoming infected with the disease. They are asymptomatic and they are potentially spreading the disease to older people who are then more susceptible. So as a result of all of this, what we're seeing throughout Europe and in parts of the United States, including here in my hometown of Newark, is curfews and closures again. And I think what we might expect over the course of the next month or so, certainly up to Thanksgiving, if not afterwards, is surgical, tailored, pinpointed closing. I don't think we can shut down the way we did before. I think there will be huge resistance to that, but we have to be methodical and thoughtful and close some things down just a little bit in order to contain the virus again until these great doctors give us the vaccine we actually need.
Thanks, Dr. Halkitis. I next want to introduce Dr. Sean Deal. Dr. Deal is an associate professor at the University of Vermont Larner College of Medicine. Dr. Deal, you recently published a study about antibodies in patients with COVID, and your study found that the patients with more severe cases actually had correspondingly higher levels of antibodies, which seemed a little bit counterintuitive. And I wonder if you can help us to interpret this a little bit and understand what that means for the body's ability to produce the antibodies against the SARS-CoV-2 virus.
Thanks for that question. And it's a pleasure to be on the panel with all these other esteemed scientists and physicians helping us get through this. So we had up at the University of Vermont, we were able to leverage our teamwork across the pathology lab and our ICU clinicians to really try to understand how quickly and the breadth of the antibody response that occurs during early COVID-19 disease. And we compared this to subjects that were actually had contracted COVID-19 but had recovered in an outpatient setting, all within roughly the same time frame. And so within the inpatient group, those hospitalized folks, the higher antibody levels were found in the sickest patients, those requiring the longest ICU admission. And those levels in general in the hospitalized folks were much higher than those than those found in patients who had recovered in an outpatient setting. And so what we think this really means is that is reflective of an arms race between the virus and the immune system. So it's really hard to accurately measure replicating virus in the upper airways in real time in a person. PCR doesn't quite get to that. That's just fragments of the virus, but the immune system really does have mechanisms in place to be able to detect ongoing replicating virus. And so we think that during this early infection stage that more antibodies are being made as more virus grows, but then as the virus gets under control, these antibodies will remain. just like in the recovered patients. And notably, the kind of antibodies we measured are those that are called neutralizing antibodies that are focused against the spike protein, that are all the subject of all of these excellent vaccine candidates that are currently in progress. So we think that this bodes really well in terms of some additional details on our study, that we found equal responses in males and females. We found robust immune responses out to 93 years of age that were similar to subjects that were infected at 6 decades younger than them. And so we really agree with the broad rolling out of a vaccine, trying to target lots of different ages and both biological sexes, and that the broad approach is the most effective one based on what we know from the live virus infection.
Thank you, Dr. Deal. I want to toss it to Dr. Spector, who wanted to add something about that immune response that you studied.
Right. No, one of the things I think that's really important is that you not only make an antibody response, but a cell-mediated immune response. And many of the people who end up being hospitalized, in fact, have an overshoot. And what seems to be getting those people with COVID-19 into the most difficulty is when you have this, what's called cytokine storm, in which, in fact, there are many cytokines that are being produced that lead to many of the complications associated with COVID-19.
Thank you, Dr. Spector. I want to go ahead now and introduce Dr. Jeffrey Shaman. Dr. Shaman is the Chief Science Officer at Coriell Life Sciences. Dr. Shaman, as we've discussed some of the vaccines and some of the understanding of the immune response, I think this kind of leads nicely to treating the people who currently have it, while we hopefully wait to understand the efficacy of these vaccines as soon as possible. Meanwhile, there's people who need treatment right now. Your company has created a DNA-based prescribing report using a genetic profile to make recommendations about what kinds of treatments can work best for certain patients. And in a case study, you guys had 2/3 of participants found to potentially need some change to their treatment. What can you tell us about this kind of precision medicine approach and how that can also be deployed against COVID?
Yeah, thank you for covering all of this, and kudos to my colleagues on the line, and for covering things like precision medicine during this difficult time. So Coriell Life Sciences uses a small sample of a patient's spit to provide insight into which medications will be safe and which will be effective for that patient. So pharmacogenomics is the medically relevant and clinically actionable science behind why some drugs work for some people and they just don't work for others. We've all heard of people that take medications that don't work for them. Many times this is because of their genetics. Additionally, we have to deal with medications that cause side effects, everything from a bellyache or a headache to severe muscle pain and even cardiac issues. By preemptively testing a person's genetics, we can really avoid that trial and error type prescribing that's often the standard course of treatment. And the risks aren't only genetic. GeneDose Live, the clinical decision support tool that we have, provides information on every prescription, over-the-counter supplement on the market in the US, and the drug-drug interaction risks, the contraindications, including those for COVID-19 disease, the FDA warnings, other medication-specific risks are there as well. So in that study you mentioned that we saw 64% of patients should make changes to their treatment, we saw a majority of those acted upon by their prescribing physicians. And this all equated to a reduction in healthcare costs, pharmacy spend, hospitalizations, and even slip and falls, and an increase in patient satisfaction rate amongst other metrics.
Thanks, Dr. Shaman. For all the media who are on the call, please chat any questions that you might have. We'll be happy to call on you and go ahead and relay those questions, or allow you to relay the question, or I'll relay it for you. I want to go back to some of the doctors working on the trials and ask Dr. Jones-Lopez and then each of the other doctors, what can you tell us about timeframes for some preliminary results or interim reporting? So what can we expect from the AstraZeneca trial in terms of when we might know more about the efficacy?
Sure. So obviously the time frame will be dependent on the enrollment period. As mentioned, two of the studies have either completed enrollment or are about to complete enrollment, the Pfizer and Moderna studies. The AstraZeneca study is scheduled to enroll about 33,000 patients, also volunteers, in about 2 months, which is basically the same target, very similar target to the other studies that I know of. And the signal, really, we're looking at both a safety signal and an efficacy signal, is going to be dependent on how quickly that enrollment period is completed. Yeah. So there's, I think, two views to this. There's an optimistic view, and then there's more of a realistic view in terms of when will we expect a sufficiently robust signal. I'm talking mostly around efficacy. And that will depend on a series of factors. The actual efficacy of the vaccine itself. We don't really know how the relative efficacy of these different vaccines. We're all shooting towards a minimum of 50% efficacy, but the variability in that efficacy estimate is going to be significant potentially. The more effective a vaccine compared to placebo, that's the other important thing to consider here. All of the studies have a placebo arm, so like a natural infection type of arm, where the rates of disease of infection will occur spontaneously as people interact in their daily lives. So one is the efficacy, the relative efficacy. The second is according to where they live in the country, the different rates of how active the disease, the transmission is occurring. I think those are the two main drivers of efficacy. The other very important signal to keep our eyes on, obviously, is safety. And the pausing of the AstraZeneca and the Johnson & Johnson vaccines are obviously around very potentially concerning side effects that are related or not to the vaccine. And that's really about, that's the reason why these pauses have occurred. And in my mind, it really just demonstrates, and this is a very important message to send to the public, it really demonstrates how the system is working as intended. And we're going through the standard safety assessments that includes the pre-clinicals that are on these large phase 3 studies that are looking at both efficacy and safety. So going back to the question, I think an optimistic view for the trials that are either complete enrollment or about to complete enrollment is a signal that is going to appear, that it's going to make itself apparent in the next 3 to 6 months after completing enrollment. And I would think that a more realistic view is maybe after that, meaning in the next 6 to 12 months, I would say. So somewhere in those two, you will probably you start seeing a signal among the different vaccines that are out there.
Thank you, Dr. Jones-Lopez. We have a question from Sam Block from The Counter. Sam, I'd love to invite you to ask the question yourself if you want to enable your audio.
Yeah, sure, I'm here. I'm a reporter at a food-focused publication. This question might be outside your wheelhouse, but I'm wondering if you have any thoughts about the recent news reports of live coronavirus being found on frozen food packaging, and I guess the related question of surface transmission.
Thank you, Sam. Any of our panelists familiar with that report or have any thoughts to share? Dr. Deal.
I could take a crack at it. Yeah, so I did heard about that report. And in my lab, we indeed study this virus and other coronaviruses as well. And cold temperature is a good way to preserve virus. And so if it's really frozen, however, a lot of things have to go exactly right for that, that non-peer-reviewed report of live coronavirus. I think that was just put out by the Chinese CDC, and then in some very strong due diligence trying to find it wherever they can. And in that case, they were able to put it into a cell culture medium, likely that is extremely conducive to growing virus. But in a real world situation, I think a lot of things have to go exactly right for that to actually lead to a case being of transmission in that case. Yeah.
Dr. Salata, I saw you gesture that you wanted to respond to this as well. Please go ahead.
Yeah, just briefly, over time and as more data has emerged, we feel that surface-related virus, which has been found, is less of a risk for transmission than through respiratory droplets or even aerosolization. And there were some recent studies just put out about finding this in sewage, that is this virus. So I think it's going to be inevitable that we're going to find it in multiple places. The relevance, the clinical and epidemiologic evidence that really that's a major threat for transmission has not been proven yet. And that's the same posture that the Centers for Disease Control has taken at this point.
Dr. Halkitis.
I just want to, I think what my colleagues have provided really remarkable information here. We have to keep in mind, though, that this is so reminiscent to me of that first decade of AIDS, for those of us who lived through it, where every single day there was a new piece of information. And I think what is challenging for so many of us doing public health work is that these reports are coming out like one after the other, and people don't know. So I'm encouraging all of us, as much as possible, especially the press, to be particularly skeptical when things come out and to present them in a manner that doesn't create panic, because that is partially what we're seeing right now in response to this disease by the human beings in our world.
Thank you, Dr. Halkitis. We have another question from Jody. She's a nurse and also writes for Quoted and for Harrow. She asks if the COVID vaccines are expected to have similar effectiveness rates as our current flu vaccines. Who can help us understand a comparison between the COVID vaccines and the flu vaccine?
So,
ahead,
it's...
Oh, sure. Thanks, Tom. So I personally don't like to compare the two vaccines, although they are quite comparable because they are two completely different viruses that follow completely different pathophysiology. But to put it simply, we hope, based on the information, we don't know the efficacy of the COVID vaccine. What it will be, that's what these clinical trials are trying to establish. And given the number of vaccines that are in development, it's likely when they're approved, there will be a slew. Some vaccines may be great in some populations and some not, and some in others. And that's why we want to make sure that all these vaccines are studied in different age groups, among different racial, ethnic backgrounds, to look at the nuances in these various age groups. Now, there are big differences with the flu vaccine. The flu vaccine and the influenza virus itself undergoes periodic changes in the viral antigens. And depending on the degree of changes, the flu vaccine that's developed every year has to adapt. And we're always playing catch up with the flu, which is why for every year with influenza, the relative efficacy of the vaccine varies. It can, in a good year, it can be as high as 90%. On a bad year, it could only be about 60 to 70% effective. But that still doesn't mean it's not effective, because even if you get the flu after getting the flu shot, you typically will have a much milder illness. We hope that with the coronavirus vaccine, that the antibody responses are robust and potent, and hopefully will not require an annual shot. But we don't know that, So these clinical trials will need to be followed to determine the type of the response and the durability of response. And let's not forget, flu has been around for over a century, right? COVID has been around for less than a year. So the information that, a month in COVID life timeline has been like light years. So the information that's provided today may be already outdated in a couple of days. So I think we have to look at the evidence and see where the science takes us with respect to the potency and the durability of the vaccines.
Thank you, Dr. Swaminathan. Dr. Spector, you wanted to add something.
Yeah, there are several points that I think are very important to understand. The first is that most of these vaccine studies are powered to show a 60% efficacy. So hopefully they'll be better than that, but they're powered to show a 60% efficacy. Keep in mind that to have herd immunity, we'd like to see at least 70%, if not higher than that. And if you look at now those adults within the US that say they are willing to take a COVID-19 vaccine study today, only 50% of people say that they are willing to take the vaccine. So the vaccine itself is not going to be something that will get us out of this pandemic. It is one tool that we will have that hopefully will decrease the circulation of COVID-19 and decrease the number of people who are becoming infected, decrease hospitalizations, and decrease those who are dying. But that needs to be combined with all the other public health measures that we are doing right now, because the vaccine is not going to be the answer. It's going to be one of the things that's going to help us get out of the pandemic.
We have a question from Samantha Black. She's from Science Board, and she asks, in a recent publication, researchers reported that antibody responses seem to last up to 5 months in mild cases of COVID. How long do the various vaccine manufacturers expect to track the long-term antibodies and protection through these clinical trials? Dr. Salata.
So in the Pfizer study, we're following volunteers for up to 26 months to look at the durability of the antibody response. And with regard to that recent publication, and that was addressed by Dr. Deal to some extent, people who had more symptomatic disease had higher levels of antibodies. And in a separate observation, some of those lasted 7 months, but there are other observations where 3 months and they're waned. But immunologists suspect that if you're rechallenged with COVID-19, you may indeed have a rise in your antibody response that may be protected. We do know about reinfection, and that's been reported in various countries now, and including in the United States. But given that 40-some million people around the world have been infected, and there are only a handful of scientifically documented reinfection cases, that still is few and far between at this point in time. And I think that's important to emphasize. A couple other points, if I may return to the influenza situation, 2 points. One is last year, the last several years, the efficacy rate has been more along the lines of 40 to 50% at best. And that depends on the age group. It's much less so in the older folks. But there again, I do agree that even if they develop breakthrough disease, despite being vaccinated, they have milder disease, are less often hospitalized, and die less frequently. So it's still worthwhile. And especially giving the flu vaccine this season in the context of the so-called twindemic is very important to understand and get that message out. However, if there's any silver lining about influenza and other respiratory viral infections, aside from coronavirus, if you look at the Southern Hemisphere, where they have seasons opposite from ours, this particular season, there were very few cases and mild cases at that. For instance, in the entire country of Australia, where they usually see about 70,000 cases annually, this was about 70. And in the entire country of South Africa, they saw 1 documented case. So all the public health measures that are being put into place that have already been mentioned and emphasized are probably very effective also against other respiratory viruses, including influenza, another one in children, although it's starting to be seen more in adults, and that's respiratory syncytial virus, and so on and so forth. So I think the measure, and I agree that the vaccine alone will not be the sole answer here. It will add to the amount of antibody responses and T-cell responses in the general population. And hopefully we'll get to that level of herd immunity that was already mentioned, but it will take a while. And we will not be able to move away from these strict public health measures in the interim.
Thank you, Dr. Salata. Dr. Deal, what would you also add to the length of that antibody and immune system response?
From the original SARS virus from 2003, which was contained much more quickly because the clinical cases were very easy to identify, but still it infected a fair number of people back then. Those individuals have been rebled this year and tested for neutralizing antibodies 17 years later after they contracted the original SARS virus, which is, as this audience will recall, is very, very related to the current SARS-CoV-2 virus that's currently circulating. And 17 years later, after exposure to SARS-1, these individuals had still retained strong neutralizing antibody responses and antigen-specific memory T cell responses that are able to be detected all this time after an infection. So we think that that's a case of natural infection. So I can't speak exactly to what vaccine will do because there was no SARS-1 vaccine that was ever rolled out. However, just based on the viral characteristics being in the same family, antigenic family, we think this bodes well for long-term protection to COVID-19.
I want to ask Dr. Swaminathan to tell us about the upcoming goal for interim analysis of the Moderna trial. And I want to work in another question from Jody, who asked a question a little while ago about whether or not the COVID virus mutating may have any impact on the vaccine.
So, Tom, let me answer the first part of your question with respect to the interim analysis. So, as has been discussed, the Moderna vaccine is fully enrolled at just over 30,000 participants. And the way the study has already designed, it was set up, was interim analyses are triggered when a set number of infections occur in the study population. So for the first, and there's two interim analyses planned for Moderna and then one a final analysis, depending on what the data show at each of those time points. The first analysis is scheduled to occur when 53 cases of symptomatic COVID infections are diagnosed within the study population. So what I mean by that is persons who are enrolled in the trial would need to have symptoms consistent with COVID-19 that will trigger them getting tested for the virus, and they have to be confirmed to have the infection to actually count for a case. And when 53 such cases accumulate in the study cohort, which we think will happen sometime in November, and that's based on when the study completed enrollment and based on the increase in cases nationwide. And at that point, statisticians will look at the data and then look at the number of cases that occurred in the placebo versus those occurring in the vaccine, and also look at the safety data. And at that time, the DSMB, the statisticians, along with the FDA, will need to make additional decisions about what to do with those data, because you still have, at that point, is the vaccine efficacious enough and has it met the threshold to really say, okay, it meets threshold for emergency use authorization. And the FDA recently provided guidance and they have released a whole document on the types of things that companies and any vaccine manufacturer would actually need to meet to be able to be eligible for EUA, for emergency use authorizations. We think that will happen in November, but the time will tell. So the timing has nothing to do with the election. It just happened to be the timing based on when the study completed enrollment to when the analysis was. And your second question, I'm sorry.
Will mutations of the COVID virus potentially affect the vaccine?
So currently we haven't seen significant changes in the COVID-19 vaccine. So as you know, most of the vaccine candidates are currently targeting the spike protein, S protein, that appears to be consistent in all the viral strains that have been studied. So will that necessarily remain true over time? Time will tell. We currently haven't seen evidence to suggest that there will be significant changes to the strains of the virus that will negate the efficacy of the vaccine candidates being studied currently.
Just one clarification. I see a chat asking about those 53 cases. I take that to mean
Total.
cases among the vaccine, the actual vaccine recipients as well as the placebo, right?
It's the entire study cohort.
That's
So
right.
that includes, because we don't know, we as the study investigators and the study team are blinded. So we don't know who actually gets the vaccine versus placebo. So that's total.
And so among those 53, if approximately fewer than 20 of the ones who get COVID got the actual vaccine,
Correct.
that would be about the threshold that you're looking for.
Yeah, it's about 31, 32 cases in the placebo to about 20 or so in the vaccine. That's the threshold. Now it could be fewer in the fewer number of infections in the vaccine in case the vaccine is more efficacious. It'll be more effective.
Thank you, Dr. Swaminathan. As we talk about understanding the vaccine, again, the current issue being the rise in cases, and as Dr. Spector put so well earlier, that the vaccine really is just one tool of many, and continuing with various other interventions is going to be necessary. I want to ask Dr. Halkitis about a study out of the University of Kansas recently that identified that counties in Kansas that implemented mask mandates had 50% fewer cases. Dr. Halkitis, is there any reason to doubt the effectiveness of mask wearing, and what would you like the public to understand about this question?
Yeah, so thanks for that question. And so as a scientist, you're always you're trained never to say 100% certain, but I will say 99.9% certain that masks are effective in a variety of different ways. The study out of Kansas parallels another set of data that have come out of Tennessee that have shown that hospitalizations in counties that have mask wearing in this second wave that we're going through are lower than in ones that do not require mask wearing. So in the absence of a randomized control trial, these naturally occurring experiments in Kansas, in Tennessee, as Dr. Salada said, in Chile and Australia, as we saw with flu season, provide sufficient indication that masks are protective. So they protect the people who are vulnerable, right? However, I believe we will ultimately find that they're ultimately protective to the person who's wearing the mask also. And so masks are effective. That is a message that is consistent. The challenge we face, of course, in our country is that the mask has become political. And when you make something that is scientific political, it loses its efficacy. But there is no doubt, reporters, mask wearing is an effective way of mitigating the disease.
Thank you, Dr. Halkitis. Dr. Shaman, I'd love to ask you about the types of patients in terms of prevention or preparedness as the pandemic spreads. What types of patients should consider getting the kind of screening and prescribing report that Coriel offers, and what that could then give advantages to in terms of treatment if they do have the misfortune of contracting COVID?
Yeah, so the CDC and Johns Hopkins guidances currently, they continue to state that there's no need to discontinue a patient's medication regimen just because of a COVID-19 diagnosis. But the importance of being on the right medication with high efficacy and no side effects is more important now than ever. But I think it's really true for medications used to treat both the symptoms of COVID, those used in the course of treating severe cases or hospitalizations, those used in clinical trials for treatment, and even those that had started prior to diagnosis. And so those include the antivirals, the antidepressants, pain management medications, anticoagulants, and even the immunomodulators. So what we know is that there's a list of indication known to increase the severity of a COVID case. So having therapeutically well-controlled chronic diseases decrease these risks. And we want to mitigate the cardiovascular challenges that are also potential side effects of some of the medications being used. So fighting the virus concurrently with a patient's existing conditions is enough of a challenge, right? Our goal, and it should be all of ours, is to improve medication management so we're not also fighting against treatment side effects and lack of efficacy. So during the pandemic, the folks that'll benefit the most from the COVID precision prescribing report are those suffering from these comorbidities, polypharmacy, uncontrolled or poorly controlled chronic illnesses, dealing with medication side effects. And I think I would add to that list something we all need to think about is those receiving treatment for anxiety and depression. But because these genetic tests help for the entire life of the patient, these genes don't change over time, anyone can benefit from these kind of precision prescribing reports.
Thank you, Dr. Shaman. I'd like to ask Dr. Salata about the Pfizer trial and what can you tell us about the timeframes for an interim analysis and what are those expectations in that study?
So thanks for the question. In the Pfizer trial, we already have sent all information along about the first 15,000 individuals that were in the study, with the next 15,000 probably by next week in their hands. They also expect, like in the Moderna situation, that they'll have information to the FDA and the independent panels by early November. And remember that Pfizer and many of the other manufacturers of vaccines were given federal money. Pfizer 1.95 billion to deliver up to 100 million vaccines by the end of December. And that is dependent, that amount of an award is dependent upon that. So they are moving rapidly. But I would like to emphasize what I think is a appropriate phrase. Everybody has heard about Operation Warp Speed. To me, that has the potential connotation that we're cutting corners, and that's far from the case with all of these vaccine campaigns. But the Pfizer CEO recently said we should move at the speed of science. And I think that's a more appropriate term as to what this is all about. So they're going to deliver, and I think they will, up to 100 million vaccines. But given that Pfizer, Moderna, and even the AstraZeneca vaccine requires 2 separate injections, that will only cover 50 million Americans. And with 325 to 330 million, assuming everyone will take the vaccine, we already spoke about that, we need much more. And I also, to end, would say we're going to need multiple vaccine platforms because of the sheer numbers and the distribution issues and everything else. So we welcome those studies with our colleagues that are doing the Moderna vaccine, the AstraZeneca vaccine, and when Johnson & Johnson is supposed to start up again this week, we're going to need multiple options, bottom line.
Thank you, Dr. Salata. Dr. Jones-Lopez, dig into that a little bit further. As Dr. Salata alluded to, one vaccine might come out and then another might then come out that's more effective, or there might be differences for certain populations. Tell us a little bit about what that means and what the public should expect with that kind of process. Dr. Jones-Lopez, are you frozen? No, there you are.
Sure. Yes, I think I'm having some internet trouble.
Yeah, you might just be
but
having
I
a little
think
bit of a
I heard
delay.
your question, which is to discuss the potential implication of certain vaccines having on other vaccines, correct?
Yeah, something coming out after an initial vaccine that might be more effective.
Yeah, so I've been thinking about this issue for some time now, and I think actually that this is probably going to be one of the most challenging elements in front of us in the next 6 months. It's going to be difficult to navigate, exactly what to do for each vaccine candidate as efficacy signals start coming out. And it's, I'm thinking of, the first 6 to 8 vaccine platforms, from the time of enrollment to the time of closure of enrollment, it's going to be roughly around 6 months. Pfizer, Moderna being the first ones that
Unfortunately,
started
Dr. Jones-Lopez,
in
we're
July, I
losing your
believe, June,
audio.
July.
Yeah.
And then the Sanofi for December. And as
I'm very sorry, Dr. Jones-Lopez. We're losing your audio, and it was breaking up there. Let me toss this to Dr. Spector. Dr. Salata, I understand, has a hard out in a couple minutes. Did you want to weigh in?
I just want to make one comment. Thank you,
Please
Tom.
go ahead. Sure.
We're not going to have comparative studies between these vaccines. So they're going to be surrogate markers, and also what was discussed by our colleague from Rutgers and others related to the antibody responses, the durability, but also those cases, true cases of COVID 19 in those that are vaccine versus placebo recipients, and the safety signals. So one thing that will happen is I don't think we should be mixing the vaccines. That's inherent in your question. If somebody starts with Pfizer, they should not move to Moderna, and vice versa, because that's not the way they're studied right now. But I think over time, we'll be able to look at the effectiveness of these vaccines, and the primary endpoint in all the vaccine studies is the number of true COVID cases between the groups. And that's going to take a while longer to accumulate that data accordingly. So I think we're going to have to start with these so-called surrogate markers, the antibody markers and the T cell responses. There'll be some data initially on the number of COVID cases between the groups, but it's not going to be such that that is the end of the story as this continues on.
Thank you, Dr. Salata. And I understand that you do have a hard out in a couple
I
minutes.
do.
So if you have to go before we wrap everything up, thank you so much for
Thank you
participating
for
today.
including me, and to my esteemed colleagues. It's been terrific. Thank you.
Thank you. Dr. Spector, what can you add on that question about an initial vaccine and then further vaccines coming out, maybe more effective or different indications?
Well, I think that there is an important population that to date has been overlooked with the vaccine. And we have to keep that in mind. There are 74 million children in the US who have not been studied. And it's only the Pfizer vaccine that has now started to go down to age 12. We know that although children seem to be less likely to get COVID-19, that they still do get illnesses associated with the infection. And we also know that they can be excellent carriers of viruses. For any of you who have ever had preschool children or school children, you know that the vector for bringing the virus into the home is often the school-age child. And so we need to be cognizant that these vaccines need to be studied in children who are less than 18, and it needs to go down to school-age children as well. Now, there's unlikely to be efficacy studies in children. And so that identifying what are the correlates of protection from the vaccine studies that are going on, so that you can now look at the immunologic effect of these correlates and how they present themselves following the vaccine in children, will be absolutely critical to expanding it to populations that are likely to be some of the most important carriers to the home of the infection.
Dr. Deal, you had something that you'd like to add on this question?
Yeah, about the outlook on the vaccines. I mean, we can kind of, as we start to know a little bit more about how the virus actually behaves, we'll be able to design better and better vaccines as we go. All of the vaccines being talked about right now are kind of version 1.0 vaccines. They're just one part of the virus, and was informed by some studies on SARS and MERS and other coronaviruses. But as we learn how to tame the virus in the lab, we can start working towards really sort of one-shot, long-term, years to decades long protection that you get with live attenuated viral vaccines. So these are likely to be a really important stopgap measure right now, because none of these technologies have ever been shown to stop a pandemic in its tracks. That's an important point. And so this is a really exciting time for vaccine development, because these kind of technologies get applied to every outbreak that happened. I was around for Zika a few years ago, and mRNA vaccines and the like were also tried. And that epidemic died out too quickly to really find out how long those immune responses last. And so, but they form really good proof of concept for being able to respond to more outbreaks, and then getting these kind of new technologies through the approval pipeline is really good from a broader sense to fight not just COVID-19, but the next pandemic that that can come as the viruses have an infinitely more higher capacity to find ways to infect humans than we currently have in the toolbox. But this response to the pandemic is going to greatly help our preparedness going forward for a lot of different things.
Dr. Swaminathan.
Thank you, Tom. So I just wanted to add, so while there are different vaccine candidates being studied, because of the investment by the NIH and the federal government, most of these clinical trials have a harmonized study design. So they have relatively overlapping, although with some nuances, inclusion exclusion criteria. They have harmonized and relatively standardized study endpoints and statistical measures to really help us be able to compare, even though the vaccine studies are directly not head-to-head. So it's not like we're comparing Moderna with Pfizer directly against one another. But the studies are being done fairly enough that you should be able to look at the results and make that decision. Secondly, with respect to endpoints, different vaccines may be better in different aspects. And what I mean by that is some vaccines may be much better at protecting severe coronavirus associated with lung infections, which typically causes a higher mortality and morbidity. It's also possible some viruses may be more effective in the upper respiratory tract and therefore have a higher impact on viral shedding and spread of infection. So they may be, maybe better used, as Dr. Spector put it, for children, having young kids, they may be the best candidates for that. And with respect to timeline, it becomes a personal decision because you try to evaluate your own risk, because if you're a first responder, if you are a person with underlying health condition or a senior citizen, you might not have the luxury of waiting for a version 2.0 vaccine down the road. You may want to get one earlier because you're trying to weigh the benefit of the vaccine versus waiting for something else better. So it ends up becoming a little more nuanced once we have more information.
I want to go to Dr. Halkitis to bring us back to what needs to be done right now and what people need to understand right now as we hold out the hope for one of these vaccines working. And I'd like to ask you if you can remind us about the importance of understanding asymptomatic cases and the pre-symptomatic phase of infectiousness. I feel like this is the thing that gets lost and forgotten about by a lot of people. And I wonder if you could really kind of put a pin in that for us.
I'll put a pin in that. I also want to just add on to my colleague Shobha's comment and just briefly say that we also remember that, again, we have lessons in the past. AIDS taught us lessons. People took medications that were not the best medications to begin with as a way of surviving, right? And the people who were sickest took them right away. And eventually we had classes of medications that are better. So for the people who need to take the vaccine, they should take the vaccine, like the first responders, like Shobha said. So back to your question. I'm sorry, I just needed to add that. Some would say we test too much. I would say that's a fallacy. We test because 40 to 50% of people who are infected with this virus are asymptomatic. And those asymptomatic people have the potential to spread the disease to those who are most vulnerable, who will become extremely sick and who will die. So given the fact that there are 40 to 50% of people who are asymptomatic out there, it is critically important that we continue to test at extremely high rates. We continue to test, to targeted tests, like we're doing at Rutgers. Our football team is being tested. The other sports team are being tested. When people come back to campus, they're going to be required to be tested. So testing is extremely important. It is often forgotten that the person who you're interacting with, who looks perfectly fine, who feels perfectly fine, may in their body, in their vessel, contain a virus who will make the person that they are talking to incredibly sick and potentially die. So keeping cautious, putting our masks on, testing, these are all strategies that are intended for the well-being of the whole society. And we have to keep doing it at these levels, and we should not deter people from testing at all.
Thank you, Dr. Halkitis. I want to ask Dr. Shaman, similarly with the kinds of precautions, somebody who might be in a high-risk population could really benefit from the type of screening that Coriell offers. How would somebody go about requesting that from their physician?
So this is not a direct consumer test. You're absolutely right. This needs a prescription like other tests out there. And I would say the best thing and the quickest is to reach out to the website coriell.com, CORIELL, to learn more, to be able to order those tests from a physician.
Thank you, Dr. Shaman. I feel like we're just about out of time. I don't want to burden these very busy doctors any longer, so you can go back to your very important work. But I want to thank you all for joining. And just a reminder to all the media who are on the call, we have a recording and we will have a transcript. If you've registered for today's event, you're going to get those things. We're going to email them to you first thing tomorrow morning. If you didn't register and you just popped in because you got a link in an invite with us or something, and you want to get that recording and transcript, shoot us an email at info@newswise.com and we will make sure to add you to that list to get that information. We'll also make sure to provide the contact details for all of the PIOs so that if you want to follow up with any of the panelists today with further questions or requests for interview or any other information about what they're working on, you'll be able to contact those PIOs to request that information. With that, I will say thank you very much to all of our panelists, Dr. Jones-Lopez, Dr. Halkitis, Dr. Shaman, Dr. Deal, Dr. Spector, and Dr. Swaminathan. Thank you all very much for participating today. Thank you to the media who joined our call, and stay safe, stay healthy, and good luck.
Thank you, Tom.
Thanks.
Thank
Be
you,
well,
everyone.
everybody. Take care.
Thank you so much. Take care. Be well.
Dr.
Thank you.
Jones-Lopez, I'm so sorry about your audio issues.
Yeah, no, I'm sorry. I'm paying a small.
Oh.